Good day, everyone. Today, we would like to discuss about the plant physiology. So first, we talk about the gaseous exchange in plants. The first question is describe the structure of a stoma. So the key point here is the structure. Okay, the structure of a stoma. So we start with a stoma is a minute opening found on leaf epidermis for gas exchange between leaf cells and environment. So we reject stomata pore because the stoma already is a pore. So each stoma is flanked by two specialized epidermal gap cells. Okay, or can say flanked by gap cells. So responsible for opening and closing the stoma. And the gap cells are associated with subsidiary cells. These subsidiary cells surround the gap cells. Okay, and the structure are different, okay, from the epidermal cells. So they are often structured different from the other epidermal cells. Subsidiary cells provide a reservoir, okay, of water and ions, okay, that diffuse into or out of the gap cell as they change shape during stomata opening and closing. The gap cell walls or the walls of the gap cells are unevenly thickened. Okay, how? The outer wall, or we call the dorsal wall, is thinner than the inner wall, which is known as the ventral wall. You must very clear. Please refer to my notes. The cellulose microfibril, they make up the cellulose wall. They arrange radially. Okay, arrange radially so that the ventral wall is less elastic than the dorsal wall. This is because when they arrange radially, most of the cellulose microfibril, they are concentrated on the inner wall. So that's why they make the inner wall becomes thicker than the outer wall. The gut cells are attached to each other at their both ends, both sides. Okay. You cannot just say attached to each other. If attached to each other, so they will combine together. Then the opening and the closing of stoma are determined by turgidity of gap cell. Okay, turgidity of the gap cell, they control the opening and the closing of stoma. So when the gap cells are turgid, the gap cells will bend outward, okay, due to uneven thickening of the cell wall, and the stoma opens. So this is what we call the structure of a stoma. Actually, we just look until this part, and then this one is just slightly, we touch slightly about the opening due to the structure of the stoma. Okay, next question, we like to discuss about the how the light intensity affect the stomata opening. So first of all, we use blue light. Okay, only blue light. Blue light photoreceptor, okay, in the plasma membrane of the gap cells are activated when they absorb blue light. The activation of blue light photoreceptors stimulates activity of proton pump, okay, in the plasma membrane of the gap cells. You must specifically mention gap cell, right? So the blue light also trigger the conversion of starch into the malic acid and then dissociate into the malic ions and proton. So this is found in the cytoplasm. The proton pumps on the membrane of gap cells actively transport. You must use actively transport. You cannot just use only transport. Okay, or you can use pump. Okay, because pump is already represents active transport. Okay, actively transport or pump proton out of the gap cell into subsidiary cell. Then the gap cell relatively becomes negatively charged. So this will establish an electrochemical gradient, okay, between the gap cells and the subsidiary cells. So resulting from the electrochemical gradient, the potassium ions from the subsidiary cells diffuses into the gap cell by facilitated diffusion. Okay, they have the potassium ion channels. Then the chloride ions diffuses into the gas cells through ion channel to balance the positively charged potassium ions. When potassium ions diffuse into the gas cell from subsidiary cells, this normally will followed by the chloride ions. Okay, then the accumulation of potassium ions, chloride ions, and the malate ions in the vacuole of the gas cells will increases the solute potential of the gas cell. Okay, or we can say the water potential of the gas cells becomes more negative lah. So you must very clear about the water potential, solute potential of the gas cell. Then water diffuses from subsidiary cells into the gas cell by osmosis. You have diffusers, you must have the osmosis. So when the gas cell becomes turgid, okay, again, like we follow the first question again, due to the uneven thickening of blah 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 blah. Okay, so the uneven thickening of outer and inner cell wall is due to the cellulose microfibers are arranged radially around the wall. So the increased turgor pressure causes the gas cells to curve, okay, or to bend outwards, and the stomach opens. Okay, so this is the mechanisms how the light intensity actually is referring to the blue light only. It affect the stomata opening. Okay, describe the mechanism of stomata closing, which is triggered by water stress. Okay, this question talk about the mechanism of stomata closing, and then this is a trigger by the water stress. So we must use the ABA. Okay, so water stress or the drought or water deficiency stimulate root and leaf of a plant to produce abscisic acid. So this abscisic acid is transported by both xylem and phloem to gas cells. Then the ABA binds to the ABA receptor in the plasma membrane of the gas cells. Okay, so these will stimulate or the binding will stimulate calcium ion channels, okay, in the plasma membrane of the gas cells to open, right? So other ion channels such as potassium ions, chloride ion, and malate ion channels are also triggered to open, okay, cause these ions diffuse out of the gas cell. So when the calcium ion channels they are open, so the influx of the calcium ion into the gas cells triggers the release of the calcium ions from the vacuole into the cytosol. So they got two ways. One is from the outside, the calcium ions influx into the cytoplasm of the gas cell. Then this will trigger the or stimulate the vacuole, okay, to release the calcium ions as well into the cytoplasm or cytosol, okay, through the calcium ion channel as well. Then the accumulation of the calcium ions in the cytosol inhibit the proton pumps. So when there are no protons are pumped out from the gas cell, so the potassium ion and the chloride ions are diffused out of the gas cells. And this is during the daytime. Okay, and again, no calcium ions can be no potassium ions can be diffused into the gas cell from subsidiary cell. So this will reduce the solute potential of the gas cell. Okay, and the water diffuses from the gas cells into the subsidiary cells by osmosis. Then the gas cells lose the turgidity and becomes flaccid, then lead to stomata closing. So you can see this, the sequence. When we talk about the mechanisms or the physiology, the importance, the sequence of the process. So like here, the gas cell, they lost the turgidity, then they becomes flaccid. Then after that, the stomata close. They cannot become flaccid first and then lost the turgidity. No, the sequence, you must very clear. Okay. So that's all for this chapter, and then please what we call continue for the next video. Okay, bye bye.